Thank you for inviting me. It's great to be in the 51st state. But it's just a joke. I want Canada to remain sovereign forever. So it's just a joke. I'm really upset about the situation in America these days. But so today what I'm going to talk about is propagating patterns of activity across the motor cortex that might signal information about movement, initiation, and movement execution. And I want to start, well, first of all, I think you asked a really interesting question last time and about this idea that there might be influences of these waves back onto neurons. I think, was it you who asked that? And I agree with John that I don't believe that's the case. I think these waves are a reflection of what's happening at this neuronal level, at the single neuronal level. They don't sort of turn back. Although, you know, I respect Earl Miller at MIT. I think he's doing very interesting work, but I don't think there's any evidence yet. If there is evidence in the future, I'm open to it. But right now, I don't believe that. So when I talk about waves or propagating patterns, well, certainly some of them, I'm just saying they're a reflection of what's going on at the single neuron level. Okay, I'm going to start with this famous map, actually, from a famous Canadian, the Penfield map, that many of you may know about. And this becomes kind of important because how do I get rid of that? This is important because it's relevant to the spatial patterns that we're seeing in the cortex, or it actually motivated me to look for spatial patterns. So you have this crude map. I guess, okay, the pointer doesn't work. But basically going from the leg, which is the most, so this is a cross section of the cortex. That's the human brain. Through the motor cortex, you've got a representation of the legs, the hand, arm, the face, and including the tongue. And this was, you know, mapped out many years ago. It's been replicated many times and is generally true. At the gross level, though, but at the finer level, it's not, it's misleading. And in fact, Penfield never made this claim. Oh, great, thank you. It turns out it's misleading because it makes you think that there's like a little thumb area right there. There's an index finger area and so forth, and a wrist area, and so forth. That is not true, actually. There is not one specific area within the arm-hand area, which is the area that I focus on, the upper limb. It's likely not true for the leg as well. And I also study the orofacial system, different parts of the mouth, the lips, the tongue. You do not find specific zones for different body parts at that finer scale. At the gross scale, it's accurate, but at the finer scale, it isn't. And so when we're looking within this sort of this localized area, the arm-hand area, it's kind of a mess. You kind of see, what you see is highly distributed representations. For example, the thumb is represented in many locations in the arm-hand area. The wrist also, and they're sort of intermingled. So there doesn't seem to be any spatial structure. And so I was like, well, you know, I'm envious of the visual people in the visual system or in the somatosensory system. By the way, there is a somatotopic map in the somatosensory system, which is much more crystalline and very clear. There is a thumb area, an index finger area, a wrist area, but that's a sensory cortex. In the motor cortex, it seems to be kind of organized in a way that doesn't make sense, or is highly, it appears random. So that kind of motivated me to look for spatial patterns. And in fact, we did find that. So actually, before I go on, I should also note that if you look, these are array recordings using Utah arrays, the same arrays that John mentioned. Two of them implanted in the motor area, the arm hand area of motor cortex. This is the central sulcus. This is the front of the brain. This is the back of the brain. So right behind this central sulcus is the somatosensory cortex. Now, there is, just so I don't make it, there is some spatial structure. So if you look here in the more medial part, you do see representation. This is based on stimulation, electrical stimulation, microelectrical stimulation of single sites. You tend to see more representations of the shoulder and elbow, more medially. And then as you go more laterally, you see representations of the wrist and the fingers. So there seems to be a proximal to distal gradient of representation. But again, there is not a thumb area, a wrist area. It's distributed and not localized. Okay, so besides this crude static somatotopic, is there any other form of spatial temporal structure in motor cortex? So over 20 years ago, well, not over, almost 20 years ago, we found found evidence of propagating waves that propagate across the motor cortex. And this is one rendition. It's smooth to make it look nice. It's measured with a local field potential. And unlike in MT, there is this very prominent oscillation in motor cortex, the beta oscillation, somewhere between 15 and 35 hertz. And so these waves are mediated by these oscillations that we see in the motor cortex. And if you look, and because there's evidence of different kinds of wave patterns in the motor cortex since the work that we did, but we've been focusing mainly on planar waves that propagate in terms of a plane in a single direction. Part of the motivation for that is that's frequently observed. It's probably the most observed type of wave we see. It's also, it seemed like a good approximation for given the size of the array relative to the motor cortex size. Unlike MT and the marmoset, which is, the array covers the entire area. In the case of these arrays, they only cover a fraction of the entire area. So to sort of a local approximation of these patterns would be a plane or a single direction. So if you look, this is now the central sulcus right here. If you look in this towards the front, so these are now, this is the front of the brain, the back of the brain is here. These are the directions of propagation that we saw in 4 different monkeys, color-coded, and they tend to propagate in one of 2 directions, either rostrally or caudally. Interestingly enough, we also implanted arrays in another structure called the dorsal premotor cortex. This is an area that's involved in the early planning of movement and less intimately involved in the actual execution of the movement. And we also see waves, but now they're oriented in the medial lateral direction. So if you now consider the entire area encompassing both motor and premotor, then you do see something that might be more complex. Although we haven't studied this yet, one could argue that the waves are propagating, this way and then changing direction. And so they're not just a single plane, single sort of direction. So they may be more complex at a larger scale, which is also something you brought up, I think. And that's something that's super interesting, at this larger scale. By the way, these are macaque monkeys, right? So I would like to do some of these experiments in the marmoset where you have a smaller brain, record from a larger fraction of the brain in the marmoset. Okay, so I'm not going to talk any more about these waves because for many years we were trying to figure out what these waves actually, what function they may serve. and we just couldn't find anything that linked these waves that we see, these propagating waves going rostral-caudal, any link between these waves and behavior. And maybe there is something there, and it's ripe for investigation. But we were struggling with trying to find what function it might play. So we investigated some other propagating patterns that are related to these waves. And the first question we wanted to address is, and we found evidence for propagating patterns that might explain this question: how come movement doesn't occur during motor preparation or action observation? So when you prepare to make a movement, you're sitting there, for example, and in our experiments with monkeys, they're sitting, their arm is sitting on a two-link exoskeletal robot with their hand on a handle. They see a cursor that they're controlling, and the cursor is at some center of the screen. They're then presented with a target that they have to reach for. But they're given we designed the experiment so that there's a period of preparation where they have to sit there. They're given the visual cue. They're told where they're going to go in the future, but wait until the target starts blinking and then initiate the movement. And of course, it's hard. It takes months to train the monkey, because of course the monkey associates hitting the target with juice, with a reward. Now you're telling them, no, you need to wait. And so during that waiting time, there's presumably preparation to move. And so we have that preparatory activity. If you record from motor cortex, you see activity during this preparation period. So how come the monkey doesn't move? Because we know the motor cortex sends projections down the cortical spinal tract that ultimately innervate the muscles. So how come you activate the motor cortex, but you don't move? That was the question, one of the questions we wanted to address. And sorry, I'm getting confused with my. So similarly, you actually see activation in motor cortex when monkeys, and for that matter humans as well, observe movements, actions. So this is a study by Dennis, former graduate student of mine, Dennis Tkach, where he had monkeys make reaching movements, but then we played back the videos, the movies, the actions the monkey had just performed on the computer screen. So they basically see a cursor moving to targets, but they're just watching it passively. And interestingly enough, we saw that in many cells, very similar modulation, like the cells responded in very similar ways when they actively made the movements versus when they observed the same movements. So again, that begs the question, okay, during action observation, how come you actually don't move? Because we actually train these monkeys to just passively watch the screen, not make any arm movements. How can they do that? Yeah.
In the training phase, is delay period.
Yes.
When they need to initiate the movement, is that always fixed, or do you vary?
In different experiments, sometimes we vary it. So we have a random period from 1 second to 1.5 seconds. And that varies from trial to trial. Sometimes we keep it fixed. But
the question is, they would learn the interval that they need.
Yes, well, they do. Yeah, they do predict. They do predict the timing of the go if you have a fixed delay. That is true. And so in that case, their reaction times would be very fast, because they'll say, oh, okay, I know when the go cue is going to come on, I start moving, and their reaction times are faster than normal. Typically, monkeys with a random interval, or even without an instruction, just see a target, make a movement. Typical reaction times are around 300, 350 milliseconds. If it's predictable, their reaction times are going to be faster. Okay, so what explains this phenomenon? you activate the motor cortex either when you plan or when you observe movement, but that you don't actually move. So there are different hypotheses. One is a gating hypothesis that was proposed by Bullock and Grossberg many years ago, saying there's something that gates the signals coming from the motor cortex, that gates the signals arriving at the spinal cord to drive the muscles. Some sort of off switch or gate. Another one hypothesis is a threshold hypothesis. Well, maybe these cells are activated, but they're not activated quite as strongly. And that, in fact, we do see that typically. Cells, when you're preparing or when you're observing action, they don't fire quite as strongly as when you're executing a movement. So maybe the cells are activated, but they haven't reached a threshold for actually triggering a movement. So downstream, there may be some nonlinearity that hasn't been reached to initiate the movement. Another hypothesis is this hypothesis by my colleague, Matt Kaufman, the null space hypothesis. And this is the idea that, well, you have this high dimensional neural state space, millions of neurons active. There are dimensions or subspaces in that multidimensional space that don't actually lead to movement. And so during preparation, you're sort of in the null space of the matrix that projects from neurons to movement. If the neural activity is meandering through this null space of that matrix, you can see activity, but no movement is generated. These are all very interesting and plausible ideas, but we had another idea. I think one thing that's missing from all these solutions is an observation we saw is this idea of desynchronization of local oscillatory activity. So I mentioned to you about the beta oscillation. The beta oscillation is this prominent oscillation. You can actually see it right here. So this is a raw local field potential trace on one trial, where 0 is when the instruction cue comes on for the animal, where the animal is given the target to reach for. There's now a preparatory period here until the go cue happens right here. And then the movement happens in this solid line. And you can see, particularly during this preparatory period, these very strong kind of sawtooth patterns, those are the beta oscillations. And these are the oscillations that mediate these waves I mentioned a few slides ago. And if you bandpass filter the signal within that narrow band of the beta, you see them very prominently. So they're very prominent and high amplitude during preparation. What's interesting, though, is after the go cue, they start attenuating. They're still present, but they're highly attenuated. And in fact, the waves that we see are still present during movement, but they're just, the signals are very weak. So here's another way to represent it. If you look at the power spectrum or spectrogram of many channels in the motor cortex during the instructed delay period, this is when the instruction cue comes on. If you see very strong beta oscillatory power, it might weaken a little bit, but it's still present there until now during the movement period. This is when movement begins. The go cue has happened somewhere before that, not clear when, maybe out here. The go cue happens, then the beta amplitude attenuates, and it remains attenuated until the end of the movement. And then it comes back on, okay? So that, and we're not the first to show this. This has been shown for decades, and it was actually shown in humans very early, soon after the discovery of EEG. So that's nothing really that new. What's new, though, actually, before I get to that, if you look at the timing at which this attenuation occurs, the timing tells you something is time locked to the movement initiation. So right here, what we did here was we took different groups of trials based on the reaction time. So these were trials where you had very short reaction times, and the yellow ones are trials that had longer reaction times. And you can see that the timing at which the beta power attenuates varies systematically with the reaction time. If you now align these attenuations to movement onset, they're all aligned. So they all, the beta attenuates consistently at some time delay before movement onset, and that's shown right in this red line. So a fixed time before the movement begins. So the attenuation of the beta might be a signal for movement initiation. Okay, so that's relatively, no one would dispute this, and it's sort of been documented in the literature before us. What does this attenuation represent? So these oscillations are occurring, then they shut off, they attenuate. Some evidence from in human studies using transcranial magnetic stimulation, they would deliver non-invasively stimulation over the motor cortex, and then at different when the oscillatory power varied in the beta frequency in the different frequency ranges, and they found that in the beta frequency range, you saw a negative correlation between the beta power, the power in the beta frequency range, and the evoked potentials evoked by TMS. Meaning, when the beta was strong, the TMS evoked potentials were small, and vice versa. When the beta power was small, the evoked potentials were large. So that suggested to me, at least, that maybe these attenuation phenomenon reflects the sort of excitability of motor cortex. So you're sitting along, preparing to move, you're at a low excitability state, and then the go cue comes on, something happens in the network and switches you to a high excitability state, resulting in low amplitude beta oscillations. Okay, so as I think John showed some examples of single trial data in the oscillations, they're kind of a mess. And if you look, although I did show you, let's see if I go back. Sorry, here's a single trial. This is a particularly good example, and you could compute the amplitude of this signal and look at when the timing occurs. But generally, they're noisy. So we had to find a way to denoise the signals. So this former postdoc of mine, Karth Balasubramanian, used an autoencoder to clean up the signals on single trials. And what I'm showing you right here is across different electrodes, each color is a different electrode, the amplitude of the beta signal, beta envelope, on a single trial. And if you look, the red electrode, which is shown here, attenuates a little later than the blue electrode, which is over here. If you now map the attenuation times on the electrode grid, you can see a propagating pattern. So it's not that like the entire cortex attenuates at the same time synchronously, but rather you see a systematic progression of attenuation beginning with these electrodes and progressing to those electrodes. This is on a single trial. On a different trial, this is now a separate trial, you see the opposite pattern. These electrodes attenuate first, followed by these electrodes. Okay, so this is something novel. that we discovered, which is the entire cortex doesn't just, the oscillations just don't attenuate synchronously, but show this propagating pattern. And in fact, if you look at many, many trials and build a circular histogram, you can see how most of them propagate in one of two directions and completely opposite directions. And it turns out this is roughly comparable to the directionality that we see with the beta waves. They're going roughly rostral, not exactly, but approximately one direction is in the more rostral direction, the other one is in the more caudal direction. And very few examples of attenuation patterns, propagating patterns in the orthogonal direction. And this is what we saw in all our monkeys. Now, sometimes we saw some subtle differences. For example, one direction was predominant, the second direction was much weaker, many fewer examples, but we always saw this sort of bimodal distribution of propagation. Sorry, I'm getting confused with these. Okay, so that's interesting. And an analogy I like to make is, and it may be the wrong analogy, but you can imagine the motor cortex sitting idly while you're preparing to move, right? And imagine you're in a car with a clutch. So you have these clutches that are down. You're pushing down on the clutch. Your engine is revving, but it's actually not moving. And then you release the clutch to engage the engine. And so the cortex, the attenuation of the beta is like a clutch being released. And so you basically have a series of these clutches that are being released in a sequential manner. That's the way I like to think about it. Okay, so can we provide causal evidence that these propagating patterns of attenuation or excitability is a necessary component for movement initiation? So we saw this phenomenon during movement initiation. It happens on every trial, but do we have any causal evidence that it may be playing a role? So what we did was we designed an experiment where we electrically stimulated, but not on the same electrode array, but stimulated with patterns of electrical stimulation. So imagine on a given trial, you have an attenuation pattern that's progressing from the right side of the array to the left side. So now imagine, so let's say on that trial, that's what's happening. And we now stimulate electrodes in this manner. So we stimulate all of these 8 electrodes at the same time with a single pulse. Then the next time point, we stimulate this column and the second column. And then progressively, we temporarily shift the set of electrodes we stimulate so that it mirrors the natural pattern of propagation that we see of attenuation. That would be called a congruent pattern of stimulation, or with the natural pattern. Alternatively, imagine we have that same pattern of propagation going from the right side to the left side, but now we're stimulating this set of electrodes first, then these 2 sets of electrodes, and these 2, and so forth. That would be incongruent. So it's going against that natural pattern of propagation. And the idea we wanted to test was, if we stimulate against that natural pattern, do we actually slow reaction time? Okay. And what's important here to note is, in both the congruent and the incongruent case, the electrodes we're stimulating are exactly the same. The currents we're using on each electrode are exactly the same, and the frequencies are the same. The only thing is the order. Did you have a question?
Yes.
Yes.
Is it still in the context where not be seen the dot?
Yes, it's in the context. So what we do is we stimulate after the go cue. We estimate when roughly when we think the attenuation is going to occur based on previous experiments.
You would not have interference of the intrinsic dynamics and what you generate with stimulation?
Well, that's the idea. We are interfering it with it. The question is the incongruent case interfering more strongly than the congruent case? But yes, we're interfering in both cases. Oh, I should also note the stimulation currents that we're using are sub-threshold. So they're low enough so they don't actually evoke a movement.
Because
Yeah. Yeah.
when you started, I was thinking that
Yes.
when you do the stimulation without the context, you would evoke a
Yes.
movement.
So yeah, sorry. If you just deliver this pattern, you will, and outside of a task, you will not evoke a movement. Okay. I mean, you might. I mean, we haven't tested it. I mean, you can sometimes measure very weak EMG muscle activity, but you don't evoke a movement. So they're subthreshold. Okay, so that was the hypothesis we wanted to test, and what we did find was, in fact, in 3 monkeys, and in one of the monkeys in 2 different movement directions, we saw a slowing of reaction time somewhere between 10 and 20 milliseconds for the incongruent stimulation pattern versus the congruent. So when we deliver that perturbation against that natural progression, we actually can slow reaction time. Not by a lot, but significantly. Did you have a question? Oh, okay. Okay. The other thing we wanted to look at and is like how do these patterns of propagation of attenuation compare to the somatotopy? So we focused. So these are 3 different monkeys. These 2 monkeys had 2 arrays. This monkey had 1 array. We only focused on these top arrays for these 2 monkeys. The blue double-sided arrow is the bimodal distribution of propagation directions of the attenuation. And the brown arrow shows you the gradient, the somatotopic gradient from proximal up here to distal up here. That is from arm and shoulder, shoulder and elbow, down to wrist and hand. And although it's not perfect, it seems that they're nearly orthogonal, these 2 different spatial patterns. The somatotopy is nearly orthogonal to the propagating patterns of attenuation, which actually, to me, kind of makes sense. If you want to initiate a movement or allow for the possibility of movement, you don't want to initiate necessarily one body part first and then another body part. You want to allow for all body parts of the upper limb to initiate if they need to. The other question we had was, okay, so this pattern that we see seems to signal a movement initiation. We had causal evidence that this pattern is necessary, maybe not sufficient, but necessary for movement initiation. How about in other behaviors? So we focused so far on planar reaching movements, but how about tongue protrusion? We actually train monkeys to stick their tongues out and apply a force with their tongue on a force transducer. We also had a precision isometric pinch grip with their hands. And we also had a human participant as part of a human brain-computer interface study I'm participating in. This human was a spinal cord injured person who volunteered to have arrays implanted in his motor cortex. But he had some residual function. He was not completely paralyzed, so he actually could make extension movements and he could apply isometric extension forces with his native limb. So in all these 4 different kinds of behaviors, we saw beta attenuation on the electrodes. And we saw them organized in a spatial way. That is, they propagated from one side to the other or the opposite. So they typically, except for in this case where the two directions are slightly off, but for the other three cases, they're nearly exactly in opposite directions, depending on the trial. Some trials they propagated one way, other trials are propagated in the opposite way, which I thought was kind of cool, even with things like the tongue. By the way, in the tongue, we're recording from the orofacial area of motor cortex, not the upper limb area. So we have the arrays implanted in a different location. Okay. So we see this pattern. The other question we had was, does this pattern tell us anything about movement, the actual movement to take place, besides the timing, besides when it's going to occur? And the answer is no, it doesn't tell us anything. I'm going to skip that. Basically, for like planar reaching movement, for example, this is the modeled von Mises function that models the bimodal distribution of the propagation directions for either movements away from the body, so like this way, or towards the body like this. And you see almost nearly identical distributions. Likewise, the speed of propagation, this is in centimeters per second, which actually matches pretty closely to what you see in MT of marmosets. You also, you see no difference in speed. Likewise, in the human subject was asked to apply isometric forces with their wrist, either at low forces or high forces, almost identical distributions, both in terms of directionality and in terms of speed. So, and we found no evidence of actual information provided by this propagating pattern as far as what movement was going to take place. So that leads me to I'm going to skip this for a second and maybe come back to it. Well, how much more time do I have?
We can stop around 12.
Around 12? Okay. I'll skip this, and then this is stuff that hasn't been published yet, so I wanted to get to it. And if we have time, I'll go back. So are there spatial temporal patterns associated with movement execution? Patterns that actually tell us something about what the movement is what kind of movement is happening. So we said, well, we have to sort of abandon the beta oscillation, and other people have looked for information in beta oscillations about movement and haven't found much. They weren't, like, for example, decoding studies where they try to decode movements from beta amplitudes. They couldn't find much at all. So we had to abandon beta, and we looked at a different signal altogether, the high gamma oscillation. So what I mean by high gamma, it's actually, it's a misnomer. It's not really an oscillation. It's power in the high gamma frequency range of the LFP, between 200 and 400 hertz. So that's quite high, and it's probably it's very close to the spiking frequency. So it's probably it's including spiking activity. And sure enough, we did this on purpose because we wanted to look at a signal that carries movement information and serves as a proxy for multi-unit activity. Something that's related to a group of neurons firing locally with surrounding the electrode tip. And previous people have used this very high frequency signal to decode movements in brain-computer interface studies. And sure enough, it works quite well. So that was the rationale for choosing this frequency range. And if you do trial average high gamma amplitudes, you can see that as you vary the movement direction that the monkey makes, you can see how the signal varies. So it carries information about what movement is going to make. So you see this very strong for movements to the left, a very strong signal, movements to the right, a very weak signal. So it seems to carry information about what kind of movement's happening. If you actually compare the tuning curves of the high gamma amplitude with multi-unit activity, because we can also record multi-unit activity from these electrodes. In fact, that's very common with Utah arrays. You're not recording from one isolated unit. You're recording from maybe a handful of units or more. This shows you the firing rate in red of the multi-unit activity as a function of target direction. And in blue, it's the high gamma amplitude. Again, matching almost identically. So across a whole population of electrodes, we see very similar tuning between the high gamma and multi-unit. So now we know what, and here I feel much more confident I know what this signal means. Whereas with the beta, it was, we said excitability, but it was, yes.
Question. The multi-unit is above how many hertz, like 500? How do you detect it?
Oh, we detected by, typically what you do is you band pass, high pass filter the signal above 350 hertz.
Okay, so you're overlapping with, okay.
You're overlapping. You're overlapping. But the way you're processing the data is different.
Yeah.
So you're high pass filtering, then you're using a threshold crossing. You're looking for spikes that cross a threshold.
Yeah.
And you're counting those
And
spikes.
the other is really just the envelope.
It's just the envelope of
Okay,
power.
so that's
Yeah. But there, yeah, there is an overlap in the frequency range. Yes. Okay, so that's trial average. Sorry. Oh, shoot. That's trial average. Beautiful. Here's single trials on the left. Each color represents a different electrode on a single trial. Kind of a mess. So again, we had to use different signal processing methods like principal component analysis first, then applied an autoencoder, then a low pass filter to get these kind of smooth single trial envelopes of the high gamma. So there's a lot of pre-processing required to get these kind of cool, smooth, nice signals. So with these signals, we could have just used the amplitudes like other people have used to decode. But actually my postdoc, Milan Rebar, had a really ingenious idea. He said, instead of looking at the amplitudes, let's look at these treat them as if they're just an oscillation. The envelopes themselves, not the high gamma, but the envelopes of the high gamma as an oscillation between 0 and 5 hertz. And let's compute the phase using, the Hilbert transform, like we did way back with the beta oscillations. So we compute the phase of each of these electrodes as a function of time, and we can lay it out on the array. So this is now at one moment in time, the phase of every electrode, but it's now the phase of the high gamma envelopes that have been low pass filtered below 5 hertz. And then you can compute phase gradients like John talked about. And from that, we can use this measure that we used in our original paper with the beta waves, this measure called phase gradient directionality, which is a measure of the degree to which these phase gradients are aligned. So basically, it's the magnitude of the phase gradient average divided by the average of the phase gradient magnitudes. And so a PGD value of 1, it would be a case where all these phase gradients are aligned, showing evidence of a planar wave. A phase PGD of 0, where they're just kind of randomly moving. The gradients are in random directions. So, yeah, a 1 is a good fit. Yeah, exactly. That's the whole point. So we can now measure the goodness of fit, of planarity, as a function of time. That's the whole point of that. So that's one approach. Another approach we took was inspired by a paper we published a year and a half ago, using a different method, a slightly different thing, but we just fit planes to the phases themselves. We didn't compute gradients. So we had to unwrap the phases. And so we now have unwrapped phase on the z-axis, and then on the x and y-axis is the phase on that particular electrode. And then just fit a plane to it. And then we can measure the goodness of fit of a plane to those unwrapped phases. And that's comparable to the PGD for the phase gradients, okay? So what was cool was using both methods, using the linear regression goodness of fit and the spatial phase gradients using PGD, what we find is a transient increase in goodness of fit starting at around 200 milliseconds before the movement begins and lasting to about 100 milliseconds after the movement ends. And that's true in both measures. If you look at the direction of propagation, now it's the direction of those phase gradients or the direction of the plane that's fit, we see comparable directions, and they're more stable across. So each black line is a different trial, I should say, and the red is the average. So each trial shows this increase in R squared or PGD, and then the average shows it more cleanly. But if you look at the direction of propagation, again, it's more consistent around this period of time when the goodness of fit is high, and they match pretty closely. Now, if you look at the direction of propagation at every moment in time, trial averaged, you get plots like this. Now, these plots are a little weird to digest. So they're polar plots where time is on the radius. So you're going from 300 milliseconds, this inner circle here is from 300 milliseconds before movement onset to 300 milliseconds after movement onset. That's the radius. And along the circle is the instantaneous direction of propagation of the high envelope, the high gamma envelope propagation. And each color represents a different movement direction the monkey was making. So this is the key to the different colors. The little arrows tell you which way the monkey was moving, up, left, right, and so forth. And what's cool is you see very similar patterns with both methods. They kind of start out very similar, and then they diverge for different movement directions, suggesting there is information there about the movement in the direction of these propagating patterns. Now, notice something interesting here. Notice these three guys right here, the blue, orange, and gray. These guys are form a cluster, and they're kind of distinct from the others. What's special about those? I was trying to rack my brain until I realized what we had done. We had overtrained the monkey. This was just a complete accident. We had overtrained the monkey to make movements out, up, or out and to the right. So this blue target, this orange target. So they were making lots of movements like this. They were highly overtrained because because of the biomechanics of the robot that we use, these kinds of movements and this kind of movement are really easy for the monkey. So these movements are harder. So he was highly overtrained for these movements. And so what ended up happening for the blue target, which the blue target end is here, he started moving towards the orange target and then switched directions. Likewise, for the gray target, which is out here, he also said, oh, okay, my default is I'm moving to the orange target. And he's like, oh shit, I got to change direction. So I'm going to move. So that might explain why these three are clustered, because the movement command, so the default movement command is very similar for those three different target directions for this monkey who's been overtrained. So again, providing further evidence that these propagating patterns tell you something about the kinematics of the movement. So we applied a very simple decoding algorithm, basically linear. It was a Wiener filter decoder to try to decode the x and y velocity of the hand as a function of time, using either the directions computed from linear regression or the directions of propagation based on the spatial gradients. And you saw very similar results. If you look at the green line, which is the composite for the both the x and y velocities together. At the beginning, the decoding is weak, and then it reaches a peak at around 100 milliseconds after the movement. And interestingly enough, it starts decreasing as the movement unfolds. Now, this is interesting to me because I'm starting to think about the motor cortex as in a kind of a different way than what people typically think. And it's the same. It's the common view is, well, the motor cortex is controlling the limb continuously throughout, and that's the basis for brain-computer interfaces, right? What we're doing is we're taking signals from the motor cortex that the subject is thinking about, and we're continuously decoding a robot device, a cursor or whatever, continuous control. But it's starting to lead me to think that maybe in the natural system, the motor cortex is actually using an intermittent control strategy. It's actually not engaged throughout a movement. It's only engaged in certain critical points. And then subcortical structures are taking over for the rest. We also, so this might be consistent with this idea that the cortex is forming these patterns, these propagating patterns. Those are mediating the commands to generate a movement, but then those commands disappear later on in the movement when these propagating patterns disappear altogether. And the continuation of the movement, by the way, continues out to at least 400 milliseconds. So it's going quite a bit beyond this point in time. This pattern, if this pattern reflects the actual motor commands from the motor cortex, they just no longer exist after that. And some other structure is taking over until the motor cortex has to. Yeah.
But brain-machine interfaces do work.
Yes.
About the Andy Schwartz
Yes.
example of the robotic arm.
Yes.
And the woman can feed herself chocolate.
Yes.
So that is, I think, reading out the motor cortex.
It is.
Signal. So is this something that you envision? It can go into a fire and forget mode, but if it needs to be more closed loop, it can engage over a longer period.
Yes. In fact, I think, and we have some data to suggest this, that in our current BCI subjects, because they can both use a BCI and use their natural limbs. So I think what's happening with BCI is they're using an quote unquote unnatural mode. They can do it, but it's not the way they normally would use their arm. They need a lot of concentration, and they have to continuously, they get into this different mode of operation. That's a conjecture. I don't have hard evidence to support that, but that's what I'm thinking is happening. Because these experiments that we just finished looking at these BCI subjects that are using their native limb, when we build a decoder with native limb movements and then apply it to the BCI context, it doesn't generalize, and vice versa. So the decoders, it's working in a different mode, I'm thinking. It's not to say it can't happen. Yes, you can get into that mode. Okay, so finally, I just want to, again, this is complete conjecture. I have no hard evidence to support this, although I know a way to actually test this. In fact, I have a grant that's hopefully, if the NIH still exists, will be reviewed to test this idea. So the basic idea is that maybe these cortical propagation patterns may facilitate recruitment order of muscles. So these are actually EMGs, muscle activity patterns for movement to the right. And you see the sequence of muscles that are activated. That's for moving to the right. For moving to the left, you see a different sequence. So if you look at like the first three muscles, for this movement, moving to the right, it's first the anterior deltoid, then it's the triceps, then it's the brachioradialis. For this movement to the left, it's first the brachioradialis, then it's the, let me think, I think it's the extensor carpi ulnaris. I may have that wrong, or anyway, our wrist muscle, extensor wrist muscle, and the third is the biceps. So maybe that's what function these cortical patterns, waves, or I'm sorry, not waves, propagating patterns might serve. And there's a way to test this because we can actually measure these propagating patterns in monkeys and also measure on each site what muscles are activated by electrically stimulating and saying, okay, are these muscles being activated in this pattern when the monkey makes this movement? And is the wave or the propagating pattern matching that sequence? So with that, I will stop. This is my lab, and thank you for your attention.
Questions? If anyone has any questions, then we'll move on to panel discussion where we can expand on the questions as well. So.
It's okay.
But I have a couple. So one of them, so when you stimulate the homunculus area